a set of genes. An operon is a set of genes that has a common promoter and operator, and the proteins encoded by these genes is responsible for the utilization of lactose. First, you have to import lactose, bring lactose into the cell. What enzyme performs that, or transport proteins, perhaps? The permease, very good. And then there's an enzyme within the cell that is responsible for breaking down this disaccharide into glucose and galactose, and that enzyme is, of course, beta-galactosidase. Now, the way we try to understand the lac operon is by looking at its different elements and seeing what particular allele we have for that element, and how, if we have two copies of that gene, then how do they sort of interact with each other in terms of how these proteins are going to be expressed or not. So let's try to understand what's going on here. You have one copy here on the bacterial chromosome, for example, and another copy on the F plasmid. So remember, the F plasmid stands for the fertility factor. The fertility factor. If we have two copies of the lac operon, then what we have created is a partial diploid. Bacterial cells are haploid, of course. They only have one copy of each gene. But by installing another copy of the lac operon into the cell, we're able to see if there is a dominant versus recessive relationship between each of these. So let's first of all look at what are the elements, right? Let's analyze each element, then try to understand how we can bring those elements together. So for the first one, we have IS, P plus, O plus, V plus, Y minus. What does IS mean? IS, remember, is the super repressor. Can anybody tell me a little bit about what you remember about the super repressor? What is the basic characteristic of the super repressor? What does it do? Things like that. What is the super repressor? Okay, yes, ma'am, it causes repression. Excellent. So what the super repressor does is that it will cause repression. It will bind to the operator and prevent the transcription of the lac operon, regardless of whether lactose is present or absent. So what is lactose? It is the inducer. What does lactose usually do? Lactose, what it usually does, for example, think of this here as a lac operon, right? Here's the promoter. What is the promoter? The binding site for RNA polymerase. RNA polymerase is the enzyme responsible for the transcription of the lac operon. Next, following that is the operator. What does the operator do? It's the binding site for the repressor, followed by the structural genes, which in our case are two that we primarily look at. The gene Z, which codes for beta-galactosidase, and Y, which codes for permease. So here's the promoter. There are two conditions, remember, that need to be fulfilled for the lac operon to be optimally expressed. One is that glucose must be absent. And the absence of glucose then will result in the high concentration of what compound? What? Cyclic AMP, very good. Cyclic adenosine monophosphate. What does cyclic AMP do? A complex with what protein? The CAP, which stands for the catabolite activator protein. When this complex forms, then what does this complex do? Binds to what? The promoter, right? It binds to the promoter. And what function does it serve there? It facilitates the binding of RNA polymerase to the promoter. So RNA polymerase on its own cannot simply go in and bind to the operator. It needs that, quote, magnet, the cyclic AMP cap complex, to be already there to help facilitate the binding of the RNA polymerase to the promoter. Once that is done, then what's going to happen? RNA polymerase is bound, then it's going to begin to move across. So then the next condition that needs to be fulfilled is what? Lactose must be present, right? Induction is a process where, in order for the enzymes to be produced, their substrate needs to be already present. Because synthesizing the enzyme is an investment of cellular resources and energy. So you want to be sure that the substrate is present for which the enzymes operate upon. If it's not present, then it's going to be a tremendous waste to produce these enzymes if they don't have anything to do. So that the next condition that needs to be fulfilled is that lactose must be present. And so how does lactose make its presence? Well, lactose acts as the inducer. The inducer will bind to, this is what's going to happen. Here's the repressor. It's going to come in, which is encoded by the I gene. It's going to come in and bind to the operator. What the effect that has is that it blocks or prevents RNA polymerase from moving across and transcribing the structural genes. The repressor acts as a roadblock for the RNA polymerase. It acts as a roadblock. But if lactose is present, lactose acts as the inducer, binds to what side of the repressor? The allosteric site. Causes a conformational change in the repressor such that it detaches from the operator, then the RNA polymerase is free to move along. But if you have a super repressor, in a super repressor you have a mutation in the allosteric site that prevents the lactose from effectively binding to it. So in other words, the super repressor is essentially immune to the presence of lactose. So regardless of whether lactose is present or absent, the super repressor will remain constantly bound to the operator and no transcription will occur because the RNA polymerase just can't get past the super repressor. So that's what you have there. P plus, that's a normal promoter, operator plus, Z plus, but Y minus, meaning that from that particular copy of the lac operon, you're not going to get anything from the Y gene. But how about the other one? On the other one, you have I plus, C plus, OC. What's OC? Correct, constitutive operator. And what's special about that, the constitutive operator? Very good. It's the super repressor nor the repressor can never bind to the constitutive operator because remember, in order for the repressor to bind to the operator, there needs to be a complementary match between the active site of the repressor and the series of bases present in the major groove of the operator. If there is complementary if the DNA in the operator has been mutated or changed such that it is now a constitutive operator, then the the repressor cannot bind to it. So the repressor nor the super repressor both cannot bind to the constitutive operator. So what that has effect is, if all else is equal, then everything after the constitutive operator is constitutively expressed, meaning it will be constantly expressed regardless of whether you have lactose present or absent. But the problem there is that the Z copy there is mutant, and the only wild type that we have is for Y. So permease is going to be expressed in both the induced and the non-induced conditions. Because Y plus is dominant to Y minus. If you have just one copy of the wild type allele, you'll automatically get expression for the permease. But how about for the Z? Are you going to get any expression for the Z? You will not, because see, for the constitutive operator gene, you have only a mutant copy of the Z gene, but for the normal one, you have Z plus. Z plus.
Can you repeat how it's positive again?
Oh, I'm sorry, could you please repeat that?
Can you repeat how it becomes positive for the permease?
Okay, why is permease positive? The reason permease is positive for both cases is, number one, you have only one normal copy of the Y gene, and that's cis to the constitutive operator. This Y gene is not going to get you anything. This Y allele is Y minus, is not going to get you anything because it's mutant. But this one, Y plus, will get you something because it's cis to the constitutive operator. It's cis to the constitutive operator, and you will get something there. The other thing is that cis and cis to the constitutive operator, regardless of whether you have lactose present or absent, you will still get a permease gene expressed. But for the Z, the problem is that the only normal copy of the Z gene that you have is cis to this normal operator, and that normal operator, when the super repressor is produced, is just going to go in and bind to that operator and never let go. So everything after that will never be expressed, so you won't get any beta-galactosidase in that case. So minus plus minus plus. Can I answer your question? And was I able to clarify a little bit?
How is it minus?
Go ahead, please. What's your question?
Can you say it again how it minus?
Okay.
Okay. So the permease, you say because the Y cis to operator?
The plus y, the y plus,
Um,
the only normal copy of the y is cis to the constitutive operator. And the constitutive operator, remember, will never be bound to the superrepressor. The superrepressor will never bind to the constitutive operator. So everything after that will be always expressed. Because remember, when the lactose is absent, the repressor is supposed to bind to the operator and not let go. And when lactose is present, it's supposed to let go. But when you have a superrepressor, it'll bind to it and not let go. But that is if you have a normal operator, if O+. If you have OC, then neither the normal repressor nor the superrepressor, neither of those can bind to the constitutive operator. They cannot. They just cannot. So when you have this clash of the titans between the superrepressor and the constitutive operator, the constitutive operator wins. But that, of course, depends on whether there are any genes following that are normal. So that's why you will get minus minus for beta-galactosidase in both conditions, because for the Z+, it's bound to the O+, which is going to be affected by the superrepressor. The superrepressor in that case can go and bind to the operator and remain bound forever. So the only copy, unfortunately, of the normal Z gene is cis to the normal operator, and the superrepressor, which is a transacting element, will be able to influence that. Remember this, that the promoter and the operator are cis-acting elements, whereas the repressor is a transacting element. What do I mean by that? Well, potentially, the superrepressor can influence not only what's here on this DNA molecule, but it can also go in and influence what's here. But the promoters and the operators, they can only influence what's following them. What's following them. And so this constitutive operator will only influence what's following it on the same DNA molecule, whereas this one will only influence what's here. How about we try the next one? A little bit more practice, I think, will help. So you have I plus P minus O plus Z plus Y minus I plus P plus O plus Z minus Y plus. So what's going to happen here? Well, again, we're going to see a similar situation. Here you have a P minus. What does P minus mean? It's a mutant promoter. If you have a mutant promoter, the RNA polymerase cannot bind to it. And if the RNA polymerase cannot bind to it, then regardless of what you have following it, will not be expressed. Will not be expressed. So we can basically forget about everything here. Let's go to what we have here. Can we salvage anything there? Well, we have a P plus O plus Z minus Y plus. Z minus over there as well. Cannot be taken advantage of because it's Z minus, it's mutant. So all we have is the Y gene again. The Y is plus, but the problem is that this O here, that's not constitutive, it's normal. So it will be influenced by whether lactose is present or absent. So in the induced state, lactose is present, and you will get expression of permease. But in the non-induced state, you will not, because lactose is not present, and the repressor will remain bound to this operator. How about for the beta-galactosidase? Well, you're not going to get any beta-galactosidase whatsoever, because the only normal copy of the Z gene is cis to the P minus. So you won't get anything there. And of course, this copy you won't expect anything either because it's Z minus. So go ahead.
When you're looking at for permease to see if it's showing you look at the Y.
Yes, ma'am, the Y. Exactly. Z stands for beta-galactosidase. Y is for permease. Very good.